Okay, 7 o'clock, I think we should start now. Welcome one and all to NTU. My name is Toh Kian Lam. I'm from the local community engagement office. Before we start, may I just request that you switch your mobile gadgets, electronic gadgets into silent mode. And of course, for emergency, there are 2 doors behind you where you came from and 2 more in front of you. Okay? Again, welcome to NTU, one of the top 15 most beautiful campuses in the world. Okay, it's true, in Asia there are only 2, NTU and Peking University in China. So, welcome because this is Friday. You should be spending your time with your family and so on, and yet you choose to come here. That means you are very, very interested in this topic, right? Yes, good. Give yourself a big hand first. Huh? Now, this is our 13th edition. We call it Did You Know Monday Talk series. We started in July last year. And one of the main purposes of doing this kind of talk is to help Singaporeans understand and appreciate research and its benefits. And every month, we normally do it on the last Friday of the month. We invite one of the top professors to talk about his or her own research area. I think that's very important. And I think it also provides a good platform for them to interact with fellow Singaporeans and friends. Now, they may be very, very famous in their own area internationally. But in Singapore, many people may not know that, right? So this is a good platform for them. And I think while you're in NTU, I do encourage you to spend some time to go around NTU to take a walk. You know, over the last few years, NTU has been transformed to probably a mini city. We have added many new buildings. We have got new dormitories, you know, the hall of residence. We have got 24 now, and we can now guarantee housing for all year 1 and year 2 undergraduate students. That's one thing. We also added in a lot of nice, iconic buildings. We have got this learning hub. We call it the Hive just next door. Take a look. We also have a new learning hub at the northern side called the Arc. We also have a very nice 90% timbered built indoor sports hall called the Wave. So there are so many things. And if you visit the North Spine, you can see all those nice eateries. We got bank, we have got travel agency, we got K-Card. You name it, you have it. So please spend some time to look around. Now for today, the program is quite simple. Our speaker will actually give a lecture. And then he will take all the questions that you have. So please ask him a lot of simple questions. I dare not say difficult questions. Yeah. So there's a flow of course, towards the end, I will also present him a souvenir. Now, the topic for today's talk is doing good with the small microbes in environmental protection and remediation. We are very, very honored to have invited Professor Ng Wen Zheng, right over here, to be our speaker. Now, Prof. Ng is professor and principal lead at Environmental Bio-Innovation Group, School of Civil and Environmental Engineering, NTU. He graduated from Queen Mary College, London University, with Bachelor of Science degree in Civil Engineering in 1977, and obtained his Master of Science and PhD degree from Birmingham University in 1978 and 1980 respectively, very, very fast. He subsequently conducted postdoctoral research on biotreatment systems at Kyoto University, Japan. Now, Prof. Ng has served as Dean, Faculty of Engineering at NUS, as well as Dean, College of Engineering at NTU. He was the founding director of Nanyang Environment and Water Research Institute for 10 years before retiring in 2017, which was last year. His research output may be found in some 600 publications. Prof. Ng has channeled his research into the design and implementation of some 130 full-scale wastewater treatment facilities via technology licensing and his spin-off companies. So he himself is an entrepreneur as well. He has also served as chairman of a major consulting firm and continued to serve as technical advisor to multinational and public listed companies operating in domains such as food and beverage, marine engineering, water and waste. And of course, he also sits on many government committees in this area. Now, Prof. Ng is also the recipient of many top awards, such as the IES Prestigious Engineering Award, Singapore Energy Award, ASEAN Engineering Excellence Award, and the prestigious President's Technology Award. He was appointed as the inaugural Tan Chin Tuan Centennial Professor in 2008, and in 2018, he was identified as among Asia's 100 leading scientists. So a very illustrious credential. So let's put our hands together to welcome our speaker for tonight, Professor Ng Woon Chur, please.
Thank you. Thank you, Ken Lam. And a good evening to you, ladies and gentlemen. I must share Ken Lam's appreciation, the fact that this is a Friday, and you are sitting here listening to a professor. By the way, to let a professor up on stage to talk is a very dangerous affair. You know, it's going to go on for hours if you are not careful. Right, but having said that, before I start with the actual subject, let me introduce myself a little bit so that perhaps you can understand from where I'm coming from. Because some of the things that I'm going to talk about may seem a little strange to you. And if you did not have some context to put it against, you may feel that research is such a directed thing, such a well-planned out thing. Of course, there is a lot of planning and there's a lot of hard work involved. But I would also like to emphasize that there's a lot in research that is due to serendipity. It is luck. All right, and good fortune that sometimes we see things that our colleagues did not see. And as a result of that, we then lead on to discoveries that we talk about, or I'm going to talk about tonight. Now, first of all, my own background. As Ken Lam said earlier on, my bachelor's degree is in civil engineering. And would you believe it? I'm actually a structural engineering specialist. So I work on concrete structures when I was younger. And because of that, if you look at my qualifications, you'll find that actually I carry a license. I'm a professional engineer, so I'm able to design full-scale systems. And that has a bearing on what I did later on in life. Now, the second part of my education was a master's that dealt with hydrodynamics and hydraulics, mathematics, all right, how we calculate things to observe their motions and all that. And that also has a bearing on what I did in later life. And the PhD, again, would you believe it, another change, chemistry. I did stereochemistry. I studied the shape of molecules. Now, this has a direct bearing on the subsequent work that I did. Very diverse. In fact, I remembered when I first came back to Singapore, to NUS at that moment in time, the university did not quite know where to put me. Should they put me in the School of Civil Engineering? Should they put me in the School of Chemistry? Somehow it didn't quite fit, and eventually they decided maybe you should go and teach environmental chemistry. Right? That was how it started. That was the beginning of my journey down the path in things environmental. And of course, given my engineering background, I went on into environmental engineering. Now, when I started, and I went around and I talked to the industry and so on, I've always been fascinated by what is happening in the industry, because the industrial folks do have lots of problems, and if we can persuade them to talk to us, you'll find that there are wonderful things that we can be looking into. Now, one of the first things that I encountered was that we had this bunch of folks. I do not want to tell you where they are from, but you can more or less guess. Right? I mean, there are only so many big operators of wastewater treatment plants in Singapore. There was this group of people who were experiencing problems with their treatment plants. And when I was brought in to look at their treatment plants, I was then brought into the plant itself and look at their reactors. Now, reactors, for those of you who are not familiar with wastewater treatment plants, reactors are essentially large concrete tanks or large steel tanks. So for the very large ones, they would typically be concrete tanks. And if you look into the tank, you'll see things bubbling away, a brownish colored water, and you think that is it. But if you were to scoop out a sample and look at it carefully, perhaps under a microscope, you would begin to see things like that. Now, if you saw something like that, then that represents a healthy population of microorganisms. So, ladies and gentlemen, we are now looking at microbes, all right? But the magnification is still not high enough for you to see the individual microbes that are in there. But this is a population of microbes. And the shape that this population has adopted, all right, so this is a shape of that collection of bacteria or other microorganisms. I would refer to it as morphology. So this is the morphology of the microbes that we have inside of the reactor. Now, if I saw this, even today, I would say, yeah, pretty good. Your plant is working okay, the microbes are happy, they're healthy, and probably they are doing what you want them to do. Now, I've not touched on what exactly we want them to do yet, all right? But they are doing things that we want them to do, and probably they are doing things in the correct way. Now, supposing now I went back into the plant at some other time, and something nasty is about to happen, and I took a sample out again, and I look at it under the microscope. I may see this, right? Now, this is an alarm going off. The morphology of the microorganisms have changed. You will notice that there are more of these strands coming out. Now, I like I'm very fond of food. I like good food. So good microbial population should look like spaghetti bolognese, all right? So you have lots of meat sauce and you have some noodles. Not too many noodles, but some strands of noodles in there. That is good stuff. But when you have too many noodles, like here, your spaghetti bolognese is going down the chute, all right? The quality is dropping, and that's not good. And if you have a cheap spaghetti bolognese, too many noodles and not enough meat sauce to hold the thing together. Now, this is the time when we panic. So now we have a problem. So that is the start of the work that I did. The question then is, why did the microbes go from this to this to this? All right? And when they get to this, the plant fails. The plant no longer is able to treat, if it's treating a wastewater, it's no longer able to treat the wastewater to the quality that we want. Now, of course, in the good old days, we were just treating sewage and throwing out the treated water into the sea. Nowadays, if we don't treat it well, it's more serious. By the way, we no longer treat sewage, we treat used water. And from the used water, we have new water. So if we messed up the treatment of the used water, we are going to mess up the treatment of the new water. And of course, that's going to have consequences later on. So we can't have that. So the control of the morphology is critical. So we need to be able to understand why this becomes this and this. And how do we stop the microbes from changing in this way? And later on, what is interesting would be, how do we reverse this back to this kind of form? That formed the basis of my work. So I've been in the business for almost 40 years. It has been a fascinating 40 years, and I spent my time talking to the bacteria, wonderful creatures. They never talk back, all right? So, you can say whatever you want to say to them, they never talk back, and if you treat them with tender, loving care, they respond, and they will do what you want them to do. Now, what is it that affected the microbes? And so here's where the research and the technology development and eventually the engineering development took place. So if I can just bring your mind back to that last slide that I had on the previous, the last picture on the previous slide. This is the picture. So we have a problem. So you have all these filaments, which I would facetiously refer to as noodles. with too little meat sauce. So how do you control these guys, change these guys back to something like that? Now, there are a number of things that we can do, and that form the basis for my research, which has continued to this very day. As time went on, first we were counting microbes, we were studying the shape of the microbes. Now we're doing DNA extraction, and we actually know exactly who is in there, and we know how many of that particular microbe is sitting in there. So the detailing has become much, much more intense compared to 40 years ago. But in those days, what we needed was, how do I control this to make sure I stay with this? And there were a number of things that, that we could do, and that was what I worked on for quite a few years. One of the first things that we discovered was the way we fed the microbes has a tremendous impact on the morphology of the population. By the way, ladies and gentlemen, if you go to a slimming center, they would always tell you, don't binge, all right? So don't have sudden starvation periods. Don't eat a lot after that starvation period. You should try to eat constantly, and you are better able to lose your weight and to hold on to the weight. By the way, I'm not saying that we are like bacteria, but actually bacteria behaves in exactly the same way, all right? If you do this fast famine style of feeding, they change, their shape change. And if you can control the ratio between the fasting and the feasting, then you can control the shape of your microbial population. Now, that aside, how do you do this control? So if I wanted to control this, if you walk into a regular let's say a regular wastewater treatment plant, you'll find that the wastewater comes in all the time, it goes through the reactor all the time, and it gets out of the reactor all the time. And there's a reason for this. Now, I don't think any one of us would like to have orders from the authorities that say that you can only go to the toilet at certain times of the day, because if you went at other times in the day and you pulled the chain and you flush, the treatment plant is not ready to receive your wastewater, all right? So the treatment plant has to be ready to receive the water 24/7. Anytime you go to the toilet, you pull the chain, it's okay, all right? The sewage will go to the treatment plant. But now I have a problem. If I did that, then I cannot control the eating that's being done by the microbes. So I need to change the control sequence that is applicable on a wastewater treatment facility. And this is where this came in then, all right? Now, most of us, when we look at things, most of us think in terms of a spatial frame. Now, what do I mean by a spatial frame? It means that in our minds, we arrange things by the location of that thing, the physical location. Like you are sitting here, you are sitting here, and you are sitting there, right? So the positions are fixed. But if I wanted to now do this sort of thing, I cannot have that. I need to change that. And that is, there is a requirement to move into what I call a temporal frame. So this is a time-based framework. Now, if I may take you one step further, if you go to your home, you can have your air conditioning units located at different places in your home. But each one of your air conditioning unit could also be controlled by a timer. All right, so that particular air conditioning unit is on a temporal frame control. But the home is on a spatial frame because you have one here, one there, one there. All right, so this was the design basis for the reactor that was developed to control this fast and feasting situation. Now, to accommodate this, then I needed to have a reactor that is different, and this is going to get real technical because that is a complete mix and an assumed complete mix. But whatever it is, I think the interesting part about it is 40 years ago, when I started my career, I truly envy my younger colleagues today. When they come in, the university will give them a big grant so that they can start off their research. They are given lots of students, PhD students, so that the PhD students can support them. Their teaching load is reduced so that they have time to write their papers and so on, all right? When I started 40 years ago as a young faculty, nothing like that. You go out and you take your chances. And we didn't even have too much money to do the research. So I'm not joking. I actually started my work with test tubes. Right? So that was how it started. There's no difference. Whether it's a test tube, a beaker, or a 100-ton tank, it is still a reactor. In my mind, it is still a reactor. A test tube is a reactor, a 100-ton tank is a reactor, a 1,000-ton tank is a reactor. The engineering is different, but the process, and especially the concept, is still the same. So that was how the work was done. It was done using a reactor that is test tube based. And I'm showing you these bubbles because I'm trying to indicate to you that that test tube could be doing different things at different points in time. There are points in time when there are no bubbles, and there are points in time when there are bubbles. Now, this is a temporal frame because a test tube did not change. But what is happening in the test tube has changed. All right? And with that, then what happens is, for those of you who are familiar with microbes, whether they're good guys or bad guys, the key thing is to be able to exert selection pressure. Stress is possibly good. Stress is not always bad. If you know when and how to apply the stress, you can create beneficial situations. And this is what is happening. This event here is creating stress. And the stress is allowing me to choose the microbes. So I'm selecting the ones that I want and eliminating the ones that I don't want. All right? So that is the basis for the reactor that came out eventually some 30-odd years ago. So, but how do you put this into a real plant? I mean, there are already sewage treatment plants 40 years ago. You know, Singapore is very advanced. We had treatment of sewage long time ago. It's not as if the moment I finish my PhD and I come back and I start talking to people, suddenly they realize they have to treat sewage. Nothing like that. It was already there, and they knew what they were doing. And what they were doing, if you look at the sewage treatment plant, so supposing now I were to send a drone over a sewage treatment plant and we took a picture, you will see blocks like that. But the key point is you will see rectangular tanks like that. So these are tanks bubbling away, all right? And then you'll see circular tanks like that. These are tanks where the water is separated from the microbes, all right? And after this, the water goes out. So this is continuous flow. How do I fit a process that is not continuous into something like that? And of course, there is a major problem, all right? You know, you have existing assets. Why would somebody allow this young PhD who has just come back and probably nobody understood what I was saying to go in there and try to redesign their plants, and worse, maybe knock down a few walls. Now, knocking down walls is really bad news, all right? Because that would damage the facilities that they have. So the solution then was to try to look for something that would allow me to design new. Now, this was one of the very early plants that I did, and it was designed from ground up, new. And if you look at this plant, by the way, this is not the plant, so this is the building that we are going to house the controls and so on. Very messy because the construction is taking place. This is the treatment facility. And you only see reactors, nothing circular, all right? Something has happened, something has changed. Now, this facility is a completely new design. 30-odd years ago, when this first came, it was considered very novel, and many people could not accept it because it was a system that did not receive wastewater continuously, but yet, in a sense, the wastewater could come into the plant continuously, but the water was not treated continuously and it was not discharged continuously. There are certain advantages associated with something like that. Number one, the reactor is a batch reactor. It has to be a batch reactor because of the cyclicity of the events that is taking place. Now, the control of the system is complex. It's not like, you know, you turn on the tap and you let the water run. This one, you have to be able to turn on the tap, you have to be able to turn off the tap, and then you have to turn it on again. So there is a sequence of events. And to expect human operators to run that kind of a plant is going to be quite difficult, particularly you're talking about 3 decades ago. So one of the first things that I had to learn was machine language, how to program the PLCs that are used, the program line controllers that are used to operate the entire plant. So it then ended up being a plant that is highly automated. Very few people. It is all automated with computers. And the plant could then run automatically, very compact layout, because you didn't need to have anything circular, all neatly laid out in little rectangular boxes. And what was unusual was that it had a very broad bandwidth for fluctuations. Now, just to illustrate this particular feature and why it is so important. 2 years ago, we were brought to Sri Lanka. Are there any Sri Lankans in the audience? There you go. And you must be aware that in Sri Lanka, in the city of Kandy, there is a very important temple, the Dalada Maligawa, which houses the Buddha's tooth. Now, that temple had a sewage treatment plant, which failed, all right? And it was not surprising. When I was brought to the temple and I looked at the plant, and I understood immediately why it failed. It failed because the temple does not have a constant population. It cannot possibly have a constant population because there are certain periods in the year where the population goes up to a few 100,000, and then it comes back down to tens of thousands because the pilgrims come, right? You know the place. The pilgrims come. In fact, I think the Perihara is about to start soon. So the population is going to go up. So whenever the population goes up, the existing plant is overloaded. It just couldn't cope with the kind of flows that are coming in. There are hydraulic upsets in the plant, and the plant failed. And when the plant failed, it was very unfortunate because the plant discharges the water into Kandy Lake, all right? For those of you who have not been to Kandy, I can guarantee you the city is absolutely beautiful, all right? And this temple and the lake is sitting right in the middle of the city. Now, here comes the nasty bit, all right? You send the untreated sewage out into the lake empties into a river, all right? And the river then takes the water to the intake point of the water treatment plant that supplies Kandy. Not good, all right? So you're really recycling the water around the lake and the river and then back to the treatment plant. So what we did was we went in and we actually designed something like this for them, and it has worked ever since. So the next time when you go back to Sri Lanka, please take a look and go, you go to the Dalada Maligawa and you can see the plant in operation down there. The temple administrator is very proud of the plant and he likes to show visitors that particular plant. Now, when you make a new invention, I think at this point, if I may digress a little bit and not just touch on the science, there's also a touch of entrepreneurship in these kind of things, all right? How we bring a new technology into the market, by the way, is not easy, all right? Why is it not easy? Because in the things that we do, first of all, we are dealing with public health. So there is built-in carefulness among the authorities and the various agencies and even the private sector. So there is a reluctance in terms of accepting things that are not well tested, all right? The other point, of course, is you are dealing with incumbents, things and people that are already in the market. And of course, they are going to resist the entry of a newcomer into the market. So typically, we will have to show, number one, we are, in terms of performance, we are at least as good or better than the previous system. And better still, we are cheaper than the previous system. So cheaper can mean in terms of capital costs. It can mean in terms of operating costs. Now, one of the first things that we noticed when we brought this thing to the market was that if you were to design and build a plant based on this cyclic procedure, based on the temporal frame, we save space. And if you just cast your mind back on the picture that was shown early on, there were two circles here, all right? If you were to design it based on the sequencing batch reactor, you don't need the two circles. So that allows the investor to save about 50% of the space. Now, some of you may think, whoa, so what? I mean, land is land, all right? Is it that valuable? Now, you try that in Singapore. It is very valuable. So the savings that we can get out of just using less land is very important. That is a huge savings. And of course, there are other things that are involved. Now, this is what a plant looks like in real life. This is arguably the largest of its kind in ASEAN. So this design came from my group, and these are the 4 reactors that perform in the way that I have indicated earlier on. Now, this particular facility is what I would call a closed-loop system. In other words, the water is treated and the water is returned to the original point where it was generated. This facility was designed and constructed for 3 things. One is it is a huge pig farm, so they were growing pigs. 2, they were slaughtering the pigs, so we had to deal with the slaughterhouse wastewater. And 3, they have a meat packing facility. So all 3 on site, and the water is being recirculated in the area. Now, you would also notice these 8 things here. All right, so there are 8 rectangular ones, and there are 8 sort of cylindrical ones. I like the number 8. All right, so I'm Cantonese, so you understand why this 8 thing comes in. So there are these 8 things here, which I will talk about later on, but I just ask you to sort of keep this in mind, all right? These 8 green things produce gas, and we want the gas for certain purposes. But at the moment, I'm focusing on this. So this is where the water is treated, and it is returned to the farm for further use. Now, I want to point out to you 3 things that I've gotten the microbes to do for us, all right? And up to now, I think I've been speaking for about half an hour or so. Right, up to now, what I've been talking about is the original work that I started, and that is getting the microbes to treat wastewater, all right? And this would be wastewater that could be sewage, right, from our toilets and bathrooms and so on. It could be wastewater from the factories. It could be wastewater from the farms, from the food preparation areas and so on. But essentially, what I'm doing is getting the microbes to treat wastewater. And the microbes are held in vessels. So these are the reactors. They could be steel tanks like this. They could be concrete tanks, and they could be very large ones, like this one here. So it's only a matter of scale when we expand the capacity of the facilities. But the concept is the same. Each one of these is based on a temporal frame, meaning time, and each one is based on using microbes to treat the wastewater. The question then is, why would the microbes want to help us to keep the environment clean? Because after all, if you thought about it, if you're talking about sewage, we are the ones who went to the toilet and pooped, all right? So, why would they want to take care of our poop? Or we went to the toilet and we peed. So why would they want to take care of our pee? And similarly, if we are looking at our factories and so on, we are manufacturing things, and as a result of that, we are throwing things out into the drains and so on. So why would the microbes want to help us to look after the environment? Now, the reason for this is, and it goes back to all things living, they want to reproduce, right? And when we design a reactor system for the microbes, essentially, we are designing them a home that makes them very, very comfortable so that they can grow and multiply, all right? And the idea is to somehow convince the microbes that whatever pollutants that we are feeding into the reactor, that is jolly good food. And they should eat it, and they should eat a lot of it. And then after they've grown, they will divide, meaning, putting it in somewhat crude terms, they are making babies, all right? So that is the whole purpose of all of this. We design a reactor, we feed the microbes so that they can make more babies. And the entire treatment process is then essentially a conversion process. We are converting the pollutants into more microbes, meaning more babies for the microbes. Now, at the end of the day, how we separate the extra microbes from the treated water is another matter, all right? So that was the basis of the work. And the early part of my work, maybe for a decade and a half or so, was how to manipulate this conversion process so that we make more microbes. The more microbes we make, the more effective the treatment process is, all right? Because you are removing more of the pollutants and converting it to microbes, and we are then taking the microbes out of the water. But something interesting happened while we were doing this, all right? We noticed that aside from producing more babies, the microbes also produce other things. For example, the fact that we are all sitting here this evening, one of the things that we are producing is CO2. And of course, I'm producing a lot of CO2 because I'm talking for the last half an hour. That is a byproduct, all right? So the microbes, when they are reproducing, they also generate byproducts. Now, these byproducts, aside from the CO2, of course, if you were to study these byproducts, then interesting things begin to surface. And that became the next part of the work that I went into. So for the last decade and a half or so, my focus has been more on this now. So I started with this, and now I'm with this, the byproducts. Now, if I now look down this side here of the slide, what are these products? Now, some of them may be byproducts, some of them may not be byproducts, but nonetheless, they are products that are produced by the microbes when they are active in the reactors. Now, some of these things, for example, catalysts. Microbes produce catalysts, and the obvious example would be enzymes, all right? Why would microbes want to produce enzymes? So for example, if you are trying to get a particular type of microbe to, let's say, attack wood, all right, lignin or cellulose, the microbe cannot swallow the pieces of wood. But nonetheless, the microbe can still degrade the wood. Why? Because it can produce enzymes. It will release these enzymes out, solubilize the lignin, and then absorb the polysaccharides that are released. Now, these enzymes are interesting. If it's not for these kind of enzymes, the whole world would be filled with rubbish. All right? It's because the microbes are producing these enzymes, that is why rubbish, or at least big parts of it, disappear. Otherwise, whatever we throw will stay there, and it will keep on piling up. So catalysts will be one of these items that we could be interested in. The second one is storage products. Now, microbes, if you push them into a fast feast, meaning that you give them a lot to eat over a short period of time, they have no time to metabolize the food, just like us, all right? So if I now sit down and have a huge dinner, not all of it is going to be productively used in terms of energy production and so on. A big part of it could be converted to fats and stored, all right? So microbes are able to do that as well. So they would be able to store carbohydrates and lipids. Now the third one, depending on the environmental conditions that we subject the microbes to, the microbes could be producing certain gaseous end products. They could be producing carbon dioxide, like us here. They could be producing nitrogen gas. Believe it or not, they can actually produce nitrogen gas if you feed them the right things. They can also produce methane, CH4. Now, this is getting interesting now, all right? So methane is a substance that carries energy. And we can use this to recover energy from the waste. The last one is even more interesting. Now, this is, in fact, totally fascinating. And this is the work that I'm working on right at this moment. Microbes produce signaling compounds, all right? By the way, you should not assume that microbes do not communicate with each other. They do. Microbes communicate with each other. They tell each other where they are. They tell each other whether they should come together, whether there's good food there or not. And they also send out signals to chase away other microbes. It's all the competition for living, good living conditions. So microbes send out chemical compounds that signal to each other. And these would be hormones. Now, what is interesting is we have found that these hormones that are produced by microbes can be mistaken by plants to be their own hormones. All right? So if I were to produce a hormone, if I were to get a microbe to produce a hormone that can be recognized by a tree, the tree will pick it up and then behave in a certain manner. Now, this is totally fascinating because now I'm jumping species. I can control another species with something that is being done on this side. So what I'm going to talk about next would be this one here and the two green ones. The one getting fat is not so interesting, all right? So we'll talk about the catalyst, we'll talk about the hormones, and very quickly and briefly, I will say a few words about the methane. Now, this is the second part of the work that was done by the group, and this deals with biogas recovery. So it's a fermentation process under anaerobic conditions, meaning no oxygen, and the microbes then have to change their metabolic pathways, meaning how they react with the compounds have to be changed. And in the process, they begin to release CH4, which is methane, and CO2, which is carbon dioxide. And of course, we can then capture this biogas and use the biogas to produce electricity, to produce steam, and so on. Now, these are some of the systems that use that concept. And typically, we would apply these on effluents with high carbon content. Now, in the region, where would this be applicable? Definitely in palm oil meal effluent. So if you were to go to, say, not in Singapore, of course, but if you were to go to, say, Malaysia, you go to Indonesia, to Sumatra, to Kalimantan, you take an aeroplane, one of those small aeroplanes with one propeller, and if anything happens, you're gone. You can fly for hours, and you still see the green stuff, all right, because that would be the oil palm trees. And if you were to land somewhere, you'll find a palm oil mill where they're milling the fruits to produce the oil. And the wastewater that comes out would go into things like that. Right? So we would do this and produce biogas. The biogas is then fed back into the mill to generate steam and electricity. And of course, in the good old days of the Kyoto Protocol, we also claim carbon credits, and we could sell the carbon credits. But of course, that market has fallen through nowadays, so that is not so interesting. So nowadays, we only get the gas, we make electricity, and we send electricity into the grid and sell the electricity that way. Now, look at some of these things here. This is for palm oil mill effluent. This is for a pig farm. This is, I think, very interesting now, hot business. This is for rubber glove wastewater. All right, so these kind of wastewaters can be found in Malaysia, for example, in Sri Lanka. Sri Lanka had a plant which I was involved with, very strong wastewaters, very difficult to treat, and yet, if you know how, you could make gas, and the gas can then be sent back into the factory for generation of electricity and so on. Now, I want to spend more time on this third piece because this is the most current piece in the work that my colleagues and I are working on at the moment. And this deals with the enzymes and the hormones that are produced by microbes, all right? We started this, as I said earlier on, because we noticed that the microbes were releasing things other than the typical things that we associate them with, all right? So the things that we typically associate them with would be more microbes, carbon dioxide, and perhaps later on methane. But now we're looking at enzymes, and more lately, we are looking at the hormones, all right? The hormones that are being generated. By the way, all these are natural, huh? So we are not making synthetic hormones. Of course, knowing the chemical structure, by the way, that's where my training in structural engineering comes in useful. It's easy for me to look at the chemical structure. And once we understand the chemical structure, we can go and do synthesis. So I could actually synthesize these things in the laboratory. But that's not the game here, all right? We don't want to do that. So we get microbes to grow, and these are the sort of things that you will see in my laboratory today. If you were to come, you see these Petri dishes, and we are cultivating different types of microbes. By the way, the colors there are natural, all right? So they represent different types of microbes, and we are feeding them with different things. For example, this one here, we are trying to get them to kill the fungus, all right? So the microbes do not necessarily live well together peacefully. They actually try to kill off each other. And if we understand which one is trying to kill off which other one, then there is scope for us to develop this into, I would say, natural pesticides. So these are sort of things that we collect from the field, and from this we would then grow the microbes. Now this is especially interesting, all right? And I would ask you to take a hard look at this, and later on I will show you other slides that will go into greater detail. Now, where would we use these sort of things? At the moment, the key application that we have found for these sort of things would be, of course, in horticulture. Right? So in Singapore, we are talking about urban agriculture, and this could be one area where we could apply these sort of things. Because with the hormones and enzymes, we could accelerate the plant's growth and also improve yield and the product quality. All right? The plantations could be using things like that, because the plantations have been applying chemical fertilizers and pesticides for a long time, and there's a lot of soil damage. So by going back to nature, so essentially we are going back to nature, reestablishing the microflora in the soil, we would reduce the use of these chemicals and so remediate the farming soils that we come across. Now, I want to take you now into the world of agriculture. I'm not an agronomist, all right? In fact, I'm a simple engineer, but nonetheless, after looking at some of these things, I think there is what I would call a deployment opportunity, meaning to go from the test tube in the laboratory out to full scale on site. And in fact, we are already doing this on site in Sumatra, in Indonesia, and in China as well. Now, what is the problem now? If you were to look at agriculture, what is happening in the agricultural world? The problem is we have been trying to intensify agriculture. Why? Because we need to feed the world. The population is increasing. So how do you feed the world? You make the plants grow faster, you make the plants produce more. And traditionally, the way we would do this is to increase the dosage of chemical fertilizers, and we increase the dosage of pesticides so that we protect the crop. However, there's a downside to all this. All this, I must say, would work, at least for a short period of time. All right? And that's why there was this increase in yields and so on, and increase in quality. But you continue doing it, then you begin to find that you keep having to increase the dosage. It doesn't end. And then you find that as you increase the dosages, the plants don't seem to be responding. And then you increase the dosage some more, the plants start to die. All right, now that's when you have a serious problem. So this is what is happening now. When we overuse chemical fertilizers, first of all, the first thing that you will notice, if you were to go and take a sample of the soil, you find that the soil is sour. It is acidic. Now, this is because the chemicals that we add causes that sourness in the soil, all right? The moment the soil goes acidic, the microbial population in the soil changes. You have different types of microbes now growing in the soil, not the ones that we want, all right? You have changed the environment that is in the soil. So you have poor microbial ecology. Now, you must never forget that the plants and the microbes work together. They are not working independently of each other. I mean, even us, all right, although, of course, every evening when we go back, we take a long shower, a lot of soap here and there, and trying to kill off all the microbes on our skin. But actually, microbes on the skin, the good ones it is beneficial. They help to protect us. Similarly, for plants, there are good microbes and there are bad microbes. And the good microbes help the plants to grow. They help the plants, for example, to grow the little rootlets. And these little rootlets are important to the plants so that it can absorb the nutrients from the soil. And this is now damaged. And once you damage this, You then have the plant diseases coming in. And I would want to show you at least a couple of these kind of diseases. And guess what? All right, when the plants fall sick, the farmers, they don't know any better, they start to pump this up. So they start to increase the pesticides that they apply, and that results in poor quality crops. They put more. And so this thing goes on, all right? It goes on, and until finally, one fine day, somebody's going to say the land is no good. We better move somewhere. And that's why we have problems with burning, because farmers need to clear new land so that they can continue growing. The existing land has been damaged. So this is why I think there is a basis, at least I believe there's a basis for the kind of work that we do. Now, by the way, by now you may have noticed that I do not just subscribe to the idea that we should do lots of research and publish lots of papers. That, to me, does not complete the journey. The journey is only completed when we start with the research in the laboratory, maybe in a test tube, and finally, I'm standing in front of a full-scale facility. That is the completed journey. So in this case, we have been working in the lab. My colleagues, and I'll point them out in a little while, have been working with Petri dishes and microscopes and all that. And now we are out in the field. And we want to see whether our ideas, the concept, can work out in the field. So this is what happens in the field when you don't do something about it. You have very poor soil texture. So if you walk to a farm and you see something like that, bad news, all right? So you are about done. And when you see this, you are done, all right? So, you know, the soil is salty and it's hard and it is crusty. Now, even when the soil doesn't look like that, if you dig into the soil, the root areas, this is what we have found, nematodes. Now, most of you will be familiar with earthworms, all right? And by the way, if you look at earthworms, some of you may get squeamish, you know, earthworms, all wriggly and so on, and you think that earthworms are no good. Actually, if you see earthworms, that is a good sign. The soil is healthy. If you take a sample of the soil and you don't see earthworms, you are in trouble. Something bad has happened. And if you stick that soil under the microscope and you see tiny little things that look like earthworms, but they're so tiny that you can hardly see them without a microscope, you have big problems. And that's where the nematodes are coming in, all right? So nematode infestation, some of you may have pots of plants at home. Suddenly you see your plant, you know, die for no apparent reason. It could be because the nematodes are chewing the roots in the soil. And once the roots are eaten, that's it. Finished. The plant dies because the plant cannot absorb the nutrients. Now, the microbes that we put into the soil, or you know, they should be there, but now we're trying to put them back into the soil, do a number of things. It improves the soil texture. Again, those of you who are into gardening will know that. You cannot have a hard-packed soil. It has to be a little bit loose so that there can be air going in and the water can flow through and your roots can penetrate into the soil. You must have a soil that does not drain too fast, because then you'll lose your water and you'll lose the nutrients, the fertilizers that you apply. Your soil microflora must be able to degrade harmful substances. You never know what is in there, so it must degrade that. And very important, it must be able to redissolve the phosphorus and the potassium. Now, a lot of folks will tell you that, oh, I need to add NPK, all right? That's your natural fertilizer. So if you go to the supermarket and you look at all the packets there, NPK this, NPK that, and so on. But in reality, there's one more element that is missing. So strictly speaking, it should not be NPK, it should be CNPK, because without the carbon, that's the C. Without the carbon, you cannot do these things here. All right? The NPK will only help this if the plant is healthy. So the idea now is to identify the useful microbes that can help in doing these sort of things, and particularly the microbes that can help to fight pathogens. There are many plants in this part of the world that are not well. They are sick, all right? And the idea is to get these microbes back into the soil so that we can increase the yields, we can increase the quality of the product. So by the way, if you see things like that, that's good, all right? So that's healthy soil. You shouldn't feel squeamish about it. Now, what is it? I want to give you one particular example on what is it that is bothering us. If you look at the picture now that is on the screen, this is called Ganoderma lucidum. For those of you who are into TCM, you may recognize this. Anybody recognize this? Lingzi, right. We eat lingzi. Lingzi is expensive. It's considered a really good herb for us, all right? But lingzi comes from a family, the Ganoderma family, that also has other relatives in there. And these other relatives really are not so nice. This one, right? Now, if you think the lingzi fungus looks beautiful, you should see Ganoderma boninense. It is absolutely beautiful, all right? It is shiny, and if you look at it, it reflects light. It's not like lingzi is a bit dull, all right? So this is beautiful, but it is deadly. I've not tried eating it yet, so I don't know whether it's deadly to us or not, but certainly it is deadly to the trees that it infects. That it infects. Now, why? Because this guy dissolves the tree, literally dissolves the tree, all right? It has got an enzyme that is so powerful that it will dissolve the tree. It's a hole in a tree, and we call that tree cancer. It literally eats into the tree. Now, I put a tick there so that you know which is a good guy, so when you go out and buy, don't buy the wrong one. The Ganoderma boninense is a fungus now that is spreading in Southeast Asia. And it attacks many trees, all right? So for example, it attacks all kinds of palm trees, woody trees, it attacks cacao, it attacks rubber, but there's one particular tree that has very significant economic value in ASEAN, and that is the oil palm tree, all right? By the way, if you have coconut trees, don't worry. For some odd reason, the coconut tree is a carrier. It doesn't fall sick. It hosts the fungus. So never have a coconut tree sitting beside an oil palm tree, because the coconut tree will then infect the oil palm tree. So you don't want that. But whatever it is, you now have a situation where this particular fungus infects the oil palm tree. Now, how should I say, the infection process is very long, drawn out. And lots of times, people may not even realize that the tree is sick unless you know what you're looking for. Now, supposing now you have the opportunity to go down to one of these oil palm plantations and you walked around and you see this, all right? By the way, if you see this, rule number one, don't stick your hand in there, because there could be snakes in there, all right? So, you have cobras and all that squatting in there. By the way, there are lots of cobras in oil palm estates because the snakes eat the rats. The rats eat the oil palm, the palm oil seed, all right? So they really get happy down there. So when you see holes in an oil palm plantation, you don't stick your hand in there. You use a stick and, you stir it up before you go anywhere near there. The other thing, of course, is that you notice that there is a gap between the roots and the soil. Now, originally, this would have grown in, but now you notice that there's a hole, and that's because the fungus is inside. All right, the fungus is eating its way into the tree. So when my team goes on field trips, one of the first requirements that we have is when we see an oil palm tree, there are certain signs. The first requirement is don't approach the tree. All right. We'll always stand a distance from a tree, walk around to see whether there's anything like that. And if there are things like that, then someone else go near and not us. So the idea is that you have to be very careful, because when you see something like that, the tree can fall over anytime. Anytime, no warning. All right, so you have to be very careful when you see this. So what are the signs that you have this Ganoderma boninense infecting the tree? So a healthy tree would look something like this. So all the leaves, the fronds are standing up, more or less, all right? As the tree gets more and more sick, the leaves sort of come down. And finally, it looks something like that. So from this stage to this stage is going to take years. Now the problem is the infection starts when the tree is still a little baby. So even as a small plant, when we transplant it out to the side, the tree may already be infected, but it shows no sign, all right? It will stay okay, it will look okay, and it will perform okay for maybe 5, 6 years. And then things like that will happen. And then after another 5, 6 years, it falls over. Right? Literally eaten through, falls over. Now, why is this time sequence important? Why am I telling you this time sequence? I'm telling you this time sequence because you need about 5 years for the tree to grow up and to start producing. And then the next 5 to 10 years is when the tree is at its peak production, all right? And then the last 5 years is when you try to decide when to chop down the tree. So this disease causes maximum damage just when the tree is supposed to hit maximum production. So it has got great economic implications when we have this kind of situation, all right? So this is not something that we want to see. And the disease is very hard to stop. Why? Because it is spread by spores. Now look at this. By the time, if you walk around the plantation, by the time you see this, so this is the fungus that is coming out from the tree trunk. So the fungus is actually in the tree, and then it comes out to the surface of the tree. Now, by the way, how the fungus travels through the tree is through the vascular system, all right? So it is like the fungus got into the roots, and then, or some wound somewhere, it goes, so supposing the tree is like us, it will get through a wound, it goes into our blood, the veins and the arteries. So it's traveling through our blood system, all right? And it's growing in there. Finally, when it's mature, when it's about ready to throw more spores out into the world, it surfaces like that. And when it's ripe, bursts, and the spores come out. So when you see this, it's actually too late already. The infection is spreading very fast. And because it is by spores, it is spread like flu. So it's very, very difficult to control. It's all over the air, all right? So this is why it is so difficult to prevent or to kill the Gardnerella. The only way is to make sure that the tree is strong so that when it's young, it's able to resist the attack of the Gardnerella organism itself and not allow it to enter into the tree vascular system. Now, this is what happens. So when we start to apply our enzymes and the hormones, you would note that I'm not telling you which hormone at the moment. When we start to apply, you'll notice that, number one, there's a difference in the soil. All right, you look at this, it looks harder. And you look at this soil, it looks looser. All right, so the soil texture is changing already. And what is more obvious is you look at the leaves, greener, stronger. And what is even more important is you look at the fruits. It's fruiting. This one is also fruiting, but it's so small you can't see it. It's inside, all right? So this is the effect. When the tree is healthy with the right microorganisms in the soil and the organisms are releasing the right enzymes and the right hormones, the tree is responding to this. And as a result of that, it grows much better than a tree that is receiving the regular chemical fertilizers and pesticides. Now, I want to show you what kind of hormones there are that play a role in these sort of things. There are different hormones, many hormones, and the hormones have particular targets insofar as the plant is concerned. By the way, these are hormones produced by the microbes, but the plant thinks that they are its hormones, so it's responding to it. So if I wanted to make the plant grow faster, I would look for a microbe that can release auxins, all right? If I wanted to have a plant that is able to resist stress, let's say drought, let's say salt, I would get one that releases abscisic acid. If I wanted something that can kill a pathogen, now I'm not telling you which pathogen, but if there is a particular plant pathogen that I wanted to kill, one of the hormones that I could apply is salicylic acid, the last one here. Right? There are other things like jasmonic acid and so on. And recently, my group has found another one, fungicide, that can kill fungus. Very powerful fungicide. Now, to do this, you now need the various microbes. And now you notice that I've not put the names of the microbes, all right? So that is IP, I'm sorry. So if you wanted to know which microbe produce which hormone, then we will need to have a separate discussion. And this, of course, is really an IP, because if you got the wrong microbe sitting in there, at best, nothing happens. At worst, the microbe may attack the tree itself, all right? So this is not something that you really want to mess about with. All right, so this is the 3 steps that we want to have. So although I started out with the functionalities, in essence, when we do the work, we start at this end here. We have to go out and hunt for the right microbes, all right? And it is this hunting business that we go out at intervals. So NTU is actually very fortuitously located. We have all the jungle that is sitting out there because of the military camp. And my colleagues go out there and collect soil samples and plant samples regularly. And we bring them back and we start to culture, all right? So we start to grow the bacteria. We separate the bacteria into different types. And we challenge the microbes to see how they respond to stress. Now, it's always about stress, all right? So we challenge them, see how they respond to stress, what kind of enzymes they would release, what kind of hormones they would release, and then we match that with the functionality that we want to have. Now, once we have that kind of a match, we then go into DNA. So we are actually working almost like molecular biologists. So you find that, my career has really taken a really U-turn somewhere along the line, which I can't even recognize now. So from structural engineering now, I'm talking molecular biology. And once we have identified, we extract the DNA, and we have identified the DNA, we now know which one is doing the job. And once we know which one is doing the job, we can then cultivate that particular microbe, grow it in large quantities, and we can then apply it on site, all right? So this is what happens in the laboratory. Now, in the laboratory is in the laboratory. Obviously, if I'm looking at an oil palm plantation which runs into thousands of hectares, I cannot be producing things in a little test tube. So this is where I would go back into the original work that I did, where I developed the reactors, all right, the cyclic reactors, which are temporal frame and so on. This is also a temporal frame reactor. So you notice that in the career that I've had, I have never quite let go of the original work that I did. That original work that was done 30, 40 years ago now is brought forward and is now being used to support the current work that is being done. Although not treating wastewater, but producing microbes that can generate enzymes and the hormones, all right? And of course, to make things cheap, we collect substrates. By the way, we don't want to say waste materials, because the moment we say waste materials, people don't want to pay us money. So we say substrates. And when you put a nice word like substrate, they say, oh yeah, this is so fancy. And maybe they'll pay us more money and support the research. So we get the waste material. We get an NPK source, very important, because we have to replace the fertilizer values in the soil, the fertility. We add in the microbes that we have grown. We select this, put all this into the reactor, we stir it, and after X time, at X temperature, and after X numbers of stirs, we get the product. And that product is what we will then apply onto the soil. This is a large-scale facility that has already been constructed. And what I want to say now is this. We started with looking at microbes to protect the environment, to treat wastewater. Then we look at microbes to recover energy, right, as in methane that is in biogas. Then we noticed that while producing the energy, the biogas, the microbes also generated different types of byproducts, enzymes and hormones. And we now realize that we can use this to support agriculture. So this is a third phase. So I started off as somebody who was very conscious of trying to protect the environment. But now my interest is food security, food safety, because we are trying to move away from the chemical fertilizers and all the chemical pesticides that are being applied. Now, just to show you the difference, These are apple trees. We did the trial in China. This is the one that is receiving chemical fertilizers and pesticides. This is the one that is receiving the bioactives, the hormones and enzymes. You can see that the leaves are greener and they're bigger. Oh, by the way, when I was there, the apples were just coming out, tiny little ones, but the peaches were ready. So I was standing beside the tree and eating the peaches. It's absolutely amazing, right? The texture and the sweetness is nothing like what we get from the supermarket. All right, bananas. I love bananas. Every morning I need to have my bananas. But I think you will realize that bananas, like oil palm, they are grown from clones. All right, so if one tree falls sick, the rest all goes, all right? And banana trees are falling sick. So there is a fungus that is attacking the banana tree. And here we have a situation where we have applied the correct hormone onto the tree, and you can see that there is a change, all right? So it's growing bananas, and the tree is looking a lot happier than what it was previously. And these are dates, a date plant, all right? Not the Middle East kind of date. The Chinese, if you are into cooking, you know, hong jiao. It's the big one, the big long ones. This one is a bit round at the moment because it hasn't quite matured yet. So again, you see what has happened when we move from chemical to the bioactives. So there's a distinct change in the yields and the quality of the fruits. Now, at this point, I think I should say a few things about, I need to do my 5 minutes pitch for the group. So these are some of the things that we have done at full scale. So all the pictures that I've shown you came from us. So all the plants were designed by us and implemented by us. All the fruit trees, we were the ones who were working with the farmers to develop those kind of solutions. So you can see all the full-scale facilities that this group has been actively working on. Some of my colleagues are here, if I may point them out, so that later on when you have questions, I can direct them to you. Sitting here is Chai Shinni. Could you raise your hand, please? All right, the young lady there, Shinni. Then Hanafi beside her. Then we have, where's Dave? I saw Dave earlier. Okay, Dave, all right. So Dr. Dave Po, he's the bioprocess person. And Dr. Pan Chao Tzu, he's the zookeeper. He's the one that looks after all the little animals that we are keeping in the laboratory, all right? So that is part of the group, and later on, please feel free to ask them whatever questions that you may have. Now, these are some of the track records that we have been producing. Of course, papers, you know, being an academic person, we have to write papers, otherwise the university would throw me out. But we have also prepared IPs, meaning patents and so on. And of course, we have received quite a number of recognition in the country. As a parting shot, the next time you go to the supermarket or to the market, take a look at some of the things that you are buying, all right? Potatoes. Look at this. This one came from this group. So I'm comparing this one with these other ones here. Look at the potatoes, the size of the potatoes. Now look at the dragon fruit. This one and this one here. Look at the difference in color. All right. Now, for those of you who are already parents, when your children play with food, be more tolerant. Playing with food is good. It's because we played with food. That's why we went down this way. All right, and we developed this kind of solutions. These are the ones that receive the hormones. By the way, in case any one of you think that, oh my goodness, you know, this bunch of scientists are feeding the plants hormones. Isn't that dangerous? I can tell you, if I were to take you there and you took a sample, unless you have very special equipment that can track hormones at very, very low concentrations, you wouldn't even know it's there. All right? The hormones are at such low concentrations, and anyway, they are naturally produced. So they are a whole lot safer than the chemical pesticides and the chemical fertilizers. Similarly, this one had the bioactives, and this one is the regular one. So the next time when you go to the supermarket, look at the dragon fruits, look at the potatoes, look at the other fruits that may be in the market, and try to decide which one is the one that had the bioactives and which one is the one that had the regular chemicals. Now, the reason why I'm saying this is these things are entering the market already. And typically, you would need to go to the more expensive shelf to find them. All right? Labeled organic, whatever, whatever. So that's where these things will be placed. With that, I thank you for your attention. Thank you.
Hello. I just wanted to ask is that when you choose the food in the supermarket, is it just go by the color to actually identify which one is with the bioactive?
Not quite, all right. Color is one indicator, and actually it may not be so easy to see because color is only one of the indicators. The other indicator is, of course, fruits that are grown this way are never perfect. So you will find some insect bites or worm bites. And that is normal because what we are applying does not kill those things. We actually encourage those things to grow. Yes.
Thank you very much. I think that's very informative. Now you mentioned about all those good things of microbes, right? So is there anything negative like using microbes in the agricultural process? I guess maybe two separate topics, how does that related to like genetic modified crops, are they totally different things or are they similar? And then my next one is more closer to home, right? So you talk about so much useful on the microbes and as a Saturday warrior, right? So a small backyard, grow things, have trees, and then if I want to do something like this, how do I get the microbes and all those? Currently what I do is that I noticed that a papaya tree, so it's have been growing quite robust. What I do is really nothing. I just put all my garden waste, just pile it on. And it seems like that's one ingredient of your process, right? So, but seems like there's more things than that, right? So, is there something sufficient, or is there some way I can acquire some of those microbes or hormones, to perfect my gardening skill?
All right. Let me take your last question first, your garden, okay? What you're doing is, in a sense, the starting point of what we are doing. Now, if you look at the hierarchy of I would say, organic fertilizers, all right? There is actually a hierarchy. At the lowest grade is soil conditioners. And you can buy this, in bags, and they're somewhat organic, peat and so on, and you go back and you apply it on the soil. So that is the lowest grade. The next layer would be the compost. You are doing compost. Whether you intended to or not, you are doing compost, all right? And in compost, you would already have some of these microbes, but it is left to chance. You are not manipulating the numbers and the type, but you are accepting natural microbes and you are allowing them to grow. And some of these may be the right ones, all right? And that's why your papaya tree is growing well. Then the next level would be what we call the biofertilizers. Now the biofertilizers are a much higher grade of compost because now we control the process very tightly in the sense that we know what is the NPK, we know what is the carbon content, we know which microbes are in there, and we also know what numbers are in there. All right? So that is a higher level. By the way, the cost structure is escalating very, very quickly now. Huh? Then the next layer is specialist biofertilizers. And these are the ones that not only fertilize, but also address pathogens. All right? So normally, your compost or your biofertilizers does not address pathogens. So the ones that address pathogens have got special microbes put into them. And these microbes will then secrete the specific hormones that attack the pathogens. The next level would then be to get the extracts. Now that is very expensive. Your compost, you spend a few dollars per kilogram, all right? The extracts will cost you a few $1,000s per gram. So it's not something that you would want to try if you're only growing a papaya tree in your backyard. All right? So that is for the specialist, horticultural people. Okay. So does that answer your question?
So how does the kitchen waste, right? So that you go to some store, the kitchen waste, and then they will give you 2 packs of them mixed in. And so which category they fall into?
Okay, the kitchen waste thing, and now there are people who are selling machines to do that. Essentially what they're doing is they are composting. They are going through aerobic composting, which, yeah, it's still composting, and that's somewhat similar to what you are doing now. Now, they may then ask you to buy certain supplements to add in, all right? You know, like instant noodle, huh? Open a packet, pour in, and 24 hours later, something happens. Now, what is in those things? There can be 2 things. It depends on the objective that the vendor is trying to get at. If the objective of the vendor is to try to degrade the kitchen waste, which is essentially things like carbohydrates, your rice, bits and pieces of meat, and so on, right? Try to degrade those things to make them disappear so that the solids are no longer there. Then much of the material that they're adding could either be an enzyme which hydrolyzes or a bacteria that releases that particular enzyme. Right, so that's what they would do. It is not so common for them to try to put in microbes that will help to fertilize or help to address some kind of special pathogens. That is a very specialized area. And typically, the people there would deal with the plantations. Yeah. And what was your other question?
You forgot already.
Yes.
I was saying that, for the microbes, so all the good things, right? Is there any negative aspect for that? I was saying that also, we talk so much about genetic modified agriculture, right? So is that a totally different topic, or that ties into what you are?
Okay. GMOs are a totally different topic. None of the microbes that we are using are genetically modified. We don't do that. There are laws against that, particularly in agriculture. So we would not want to run the risk of the agricultural products being rejected because we applied a GMO on the microbe. And it's because we don't apply GMOs, that's why my colleagues have to go hunting. We have to go out to the natural environment and try to find the natural microbe and bring it back and try to work out how to cultivate the microbe. By the way, growing microbes is not easy. They're quite finicky. Some of them, they don't like this, they don't like that, they die, and so on and so forth, all right? So being able to grow them is an art, and that's where my colleagues come in. So just to reassure you, at least us, we don't deal with GMOs. It's a separate issue altogether. Okay?
Great lecture. 2 things. First, I presume when you talk about bioactives and you're scaling up to a plantation level, you must be having some form of self-replication. In other words, there will be some form of soil treatment that actually you can't be doing it for every tree and every season, every tree, right? So I presume that's what you're doing.
Yes.
Yeah. Then just a comment about following on the previous Q&A. I think maybe there is a market for Singapore in terms of HDB or horticulture. We know that ideally you should have earthworms to go and help aerate your soil. But if you're talking about the HDB corridor and the little guy's planter box somewhere, you're not going to have that.
Yep.
So maybe although it would be high value, it might be worthwhile actually getting some form of conditioners so that people can enjoy the indoor gardens, have potting mix that works. I think if your hobby gardener can't get success, you don't go further than that. But if you change the culture, I think that is your next change. You've changed a lot of paradigms for Singapore, I realize. But I think that might be the next paradigm you might change. Thank you.
Yeah, I'm very, very conscious of the fact that we are trying to get into a sustainable city situation. And sustainable city must mean that we have good living spaces, we have enough water, and very importantly, we must have enough food. No point, saying we are sustainable and the moment somebody turns off the taps and we are in trouble, all right? That doesn't work. Now, I take your point on the HDB estates, whether it's planter boxes or on the roof. We're thinking about the roof, and the fact that we may need earthworms, all right? Now, let me take you down this path of reproduction again. There are people in Singapore that are already producing vermicompost, meaning compost that is produced by worms. So the worms eat the stuff, they digest it, and then they secrete it, and you use that mixture, the secretion and the worms, to grow your plants. It works beautifully. And of course, anybody can go and buy a bag of that and put it in the planter box or even on the gardens on the roof. Why is it that we don't do this when we go to the plantations? There has to be a reason, all right? And the reason goes back to at the very beginning of my talk, reproduction. For things to be scalable, we must be able to do things fast, meaning the turnaround has to be very fast. Because if it takes a long time, then the systems get very big, and with size, the cost gets very high. The fact of the matter is, although you may think, or we may think here, that earthworms lay a lot of eggs, and they do lay a lot of eggs, and so they reproduce a lot. But if you were to compare the reproductive rate of the earthworm and the bacteria will beat the earthworm anytime, right? And that is why we focus on the bacteria. The fungus is also fast, but there are problems with fungus, all right? So we focus on the bacteria. And for large-scale application, we don't use the earthworms. But certainly in your application, I would say the earthworm is something that...
Sorry, the point I was going to make is that most people don't really like the idea of earthworms. So you would have something that you can buy in the shop and then do the job. Yeah, I think this is a matter of education, all right? We have to educate our people that earthworms are good guys, all right? It's when you don't see earthworms, then you should start to get really worried. It's just like, when we go to the market and we look at a fruit and you see some insect has bitten it off, I would say, I want that one because the insect didn't die, all right? Yes.
Evening, Prof. Ng. Just wondering, right, for your microbe, the hormones, right? Have you tried to apply at municipal sewage sludge, to convert into fertilizer? Because I think Chinese, they try to do it, but I think there's a lot of complication, heavy metal, that kind of things.
Yes.
So what's your view? Can you do it better?
No, because the start material is very important. You cannot change the start material, especially if it is metals. The problem with sewage sludge, and it's not just a Chinese, almost everybody else's sewage sludge is the same. There are always metals in there. And if you use that sludge and we can make this material, all right? We can grow the microbes, and the microbes will produce the hormones. The problem is when you apply this onto the soil, the metals will leach out, and the plants will soak up the metals, and guess what? The metals will go to the fruits or the tubers. So these are the two ends of the tree, right? The fruits or the tubers. And we eat the fruit or the tuber. So we are not doing ourselves any favor if we do that. What we particularly like would be agricultural waste. All right, so when you harvest things, you only take a little bit. Let's say you go and pluck an apple. We take the apple, but the leaves and the branches we throw away. So what do you do with that? Now that is what we want. We'll use that to make all these things.
I'm just wondering.
Sorry. I'm sorry.
Hello. Sorry. I'm just wondering. You mentioned the microbes in the soil. I wonder, the microbes, do they live also well just in pure solution? The environment that provide to them, does it have to be soil based, or could it be just water based or solution based?
When we grow the microbes in the lab and we harvest the microbes, we actually freeze dry the microbes. So we keep the microbes like a powder in the cold store. All right? So that allows us to keep the microbes for the longest time. The moment we mix the microbes with any solution, water or water mixed with something, the microbes will start to revive. And the shelf life of the microbes will then start to drop. So one of the key things that we must always remember when we are dealing with microbes is to keep things dry. All right.
what I mean more specifically, I'm referring to, for example, hydroponic system. So that's completely soilless system. Now, will it be equally good that we
would the
use
microbes grow there?
into the
Yes.
solution for microponic system?
It would work, yes. The microbes would be able to live in there. Provided that which I think it would be, provided that the water medium has got oxygen, which I think you would have. Yeah, in hydroponics, you would actually inject some amount of air, and we need that to keep the microbes happy. The nanobubbles are to allow you to transfer oxygen into the water more effectively. It does not quite, I mean, there's an influence, but it's not a direct influence on the microbes. That saves your operating costs, because when you transfer the oxygen more effectively, you use less energy. That is the reason why we use nanobubbles. Yes.
When you first apply the bioactive ingredients into the soil, do you need to apply again after after they have used it up?
We do.
Or when is the next application?
It depends on the crop. All right. Some of them is it depends on the life cycle of the tree. So if your plant has a long life cycle, say in terms of 10, 15, 20 years, annually it could be 2-3 times. If you're looking at a crop, let's say, in a few months, you would finish and you would harvest, then it could only be one time. So there's no hard and fast rule, crop dependent.
Well, that means if it is a durian tree, which will live to about 100 years, then we have to apply annually.
More than annually. Do you have a durian plantation? I would love to go there. You have one? Then if you have a durian plantation, I wonder have you examined the trees? Do you have any tree that is suffering, there's a colloquial term for it, bleeding heart? Right. They may be suffering from bleeding heart. That is a fungal attack. All right. So if that is the case, then what we do can be useful, because the hormones that we are producing attacks fungus. And of course, the durian is of not recently, but usually the durian is of high enough value for you to consider things like that. Papaya maybe not so good. Yeah. Yes.
Hi, I have a few questions. Thanks for sharing, because I totally know nothing. The first question is, how fast does it take to see results when you apply the good microbes to those problematic cases?
Months.
Months?
Yeah, this is living. It's not like a chemical. You cannot just put it there and then tomorrow something happens.
Right.
You put the microbes in, the microbes need to revive, they need to multiply, and then they need to release the hormones, and then the tree needs to absorb the hormones. All this needs time.
So as far as adoption is concerned, how is it adopted in the deployment? Are people keen on such solutions when you try it?
We are now working on trials, large trials. It's still fairly new, especially on the pathogen situation.
Right.
We'll know in about 3-4 months' time.
I see. So does NTU have startups that commercialize this kind of deployments?
Maybe we should talk after
it's
this.
Very
yes.
interesting. Yeah.
Yes.
Because I wonder if it's just limited to academic research and
Oh,
deployment.
no, most certainly not. I would be keen, if you are interested in investing in a startup, small money, we can talk afterwards, yes.
It's very interesting.
Yeah.
The last question is really to understand what are your future plans in what other areas are you looking at, in future?
Oh, I think the focus will still be pretty much on pathogens, because we are now discovering more and more of the food crops that we are growing are suffering from pathogenic attack. So even the durian has that attack. All right? I call it the bleeding heart because the tree has a sort of a cancerous growth, and then the red sap comes out, bleeding heart. Rice has a problem, there's blight. Bananas have a problem, there's fusarium. Corn has a problem, there's another blight there. So I think just working on these things will take the group. I only have a very small group. I've already identified, that's the whole group. It's going to take us a while to work through all of this. It's not easy. We have to go out, we have got to look for the microbes, we come back, and there will be cases where we find nothing useful. And then we have to go out again, because we have a point that we will not do GMOs. All right? And because of that, the whole process is very long, drawn out. Everything has to be naturally out there, and we hunt for them. Anything else? Thank you.
Hello. About the plantation part, that the soil condition for each country, for each place will be different. Does it mean that your solution will be only applicable for one plantation after your study, or it can be generalized to one type of plant?
Okay. It is generally applicable for a particular type of crop, all right? But having said that, the soil conditions do have an impact on the microbes that we are applying, because they are expected to live in there. So typically we will try to match it with the crop, and then we have to study the soil to match the soil as well. And there can be some differences. But we're talking about a population. So the bulk of the members of the population would be applicable. But there will be certain members that we may have to change because of the soil conditions. All right? Something else. Because we are working with pathogens, we are not like some of the other folks. For example, they work on nitrogen fixation and so on, or solubilization of potassium and phosphorus. We do that, but our focus is not that. Our focus is on pathogens. And the strain of the pathogen plays a role. All right, so just because it works on the pathogen here doesn't mean that it will work on the pathogen there, although they may look the same, but it's a different strain. So because of that, we also have to modify the strain that we are applying on the field.
So in this case, you will have different solutions for different case, more or less.
Conceptually, the solution is the same, but the specifics can be different, yes.
Okay, then in this case, you are actually changing the macros in the soil in terms of the population-wise. Have we considered about the interactions with other macros or other things in the soil or other things in the plantation? Will that impact, other things get impacted?
It would, but and there was a question, would what we are doing affect the ecology of the area? And the simple answer is, if you overdo it, the answer is yes. It would affect the ecology. And we are very mindful of that. But having said that, if I may sort of defend the work that I'm doing, the reason why we appear there is because the ecology is already damaged. Otherwise, we wouldn't be in there. So if you took a sample of that and you look at the ecology, it's gone. The insects are gone, the worms are gone, and now we are trying to put them back. Of course, the counter-argument is, are we putting back the original microorganisms? We can't do that. There's no way we can do that. So we do what is next best, and that is we are reintroducing natural microbes back into the environment.
Okay.
No GMOs.
Yeah. Thank you.
Thank you.
Just we always talking about material organisms. Have you all using this microbead process to treat those industrial polluted type of soil? That means on the soil remediation aspect. Can your this microbead to handle to revitalize, like for example, a lot of cyanide all these industrial waste, chemical waste, and all this on our ground condition? Which is because I heard there's some other technology, they inject nutrient into the soil condition and then they can neutralize it through a period of time. Is it yours or similar in this aspect?
Yeah. Similar in concept, but in the specifics, not similar. Because that would require specific types of microbes that can degrade, for example, cyanide. We are not looking at that. We are looking at microbes that can attack pathogens. So if we were to apply that kind of microbes on our kind of application, it would not work, and vice versa. But the concept is the same.
So you had no intention to look into those that type of waste, badly polluted type of soil remediation aspect?
Oh, we have the intention, but we don't have the money. So
okay, then we can discuss.
Okay. Very good, please. Because research requires funding, unfortunately, and we can only do so much at a time. Now, what we have done, by the way, is not so much directly on the soil, but because you are talking about contaminated soil, so I would presume that you're also perhaps interested in things like contaminated rivers and lakes and so on, right? What we have done is to look at the plants that grow in these kind of conditions and what we can do to enhance the growth of the plant. Because the faster the plant grows, the faster the decontamination will take place. All right? The plants absorb the metals and the various pollutants. We did that in Sri Lanka, in Candy Lake.
This is just a question about the paddy cultivation. Now, I'm referring to Sri Lanka. Most of the paddy fields are, when the salt water comes into the paddy fields, and they are not. You cannot continue in there. So, do you have any solutions to address issues like that to reduce the salt content?
Okay. We have found a hormone that will increase salt tolerance. All right. But having said that, please don't try to grow your paddy with seawater. It's not going to work. But if your soil is contaminated with some salty water, then this hormone will increase the paddy plant's tolerance to it. Yes.
So any last question from the audience? Well, if not, we thank you for your active participation. Very good questions. And we also must thank Professor Ng for such an insightful sharing with all your, I'm sure you get all your answers, right? And same as you, he actually sacrificed his Friday night to review. And it's expensive, but he did it for free. This is part of the community engagement. So you cannot charge me now, huh?
And I've not eaten dinner yet, and he's trying to avoid buying me dinner.
So again, let's please give Prof. Ng a round of applause. Yeah. And of course, I'd like to present Prof. Ng a small token of appreciation. Here you go.
Thank you.
Thank you so much.
Thank you
so much. Thank you so much.
Thank you. Thank you.
And for those who are taking buses, I think it's 9 o'clock. Yeah, so just proceed to the way you actually alighted just now, and then take your bus. All right, and have a wonderful weekend. And if you have any questions, stay back, talk to Prof. Ng, gentlemen here. You got some investment interest, please. And upstairs, the lady as well. This like what? Business pitch or what? I don't know.